Hello and welcome to the World Hatchery Forum, a webinar series focused on practical solutions for hatchery managers. Through 4 webinars, the series will discuss fish and shrimp feeds, genetics, as well as recent equipment and technology innovations. My name is Lucia Barreiro, editor of Hatchery Feed and Management, and I will be your moderator. Today is the 3rd in the series that will focus on fish hatcheries and will discuss nutritional solutions for early fish stages and genetics. Today's webinar is sponsored by BioMar and INVE Aquaculture. Each speaker will give a 15-minute presentation, and then we will get to a final panel session to answer questions from the audience. To submit a question, please type it in the Q&A box. Special thanks to our assistant editor and webinar manager, Marisa Yanaga, for making all things work. The panel today was announced to be of 4 speakers, but Björn Ronch is not able to attend. But we still have 3 great speakers and presentations. And let's start with the first one, Joana Amaral, who is BioMar's Global Product Manager for Marine Fish Hatcheries. Joana manages and develops the marine fish larviva product portfolio at a global level. Joana holds an MSc in aquaculture by the University of Stirling. Before joining BioMar, he gained solid knowledge and hands-on experiences in the hatchery and the marine RAS segments. She was hatchery manager for more than 20 years, working for different private companies in Portugal, Cyprus, the UK, and Spain, with different marine fish species, always striving for improvement. The title of her presentation is Best Start in Life. Joana, hand it over to you.
Thank you, Lucia, and good morning or good afternoon to everyone. Let me share my screen. I suppose that you can see my screen now.
Yeah, it's okay.
Okay, so today I want to talk to you about, like Lucia said, about the best starts in life. When we talk about marine hatcheries, we're obviously talking about broodstock, we're talking about larval rearing and juvenile production until fry. And the main objective of any hatchery, it's actually to be producing quality, robust and performing fry for the grow out stages, to supply and to have a positive impact and work well in the grow out stages. Of course, we want to do that with a good biological performance. So we're focusing on survival, growth and deformities. Having achieved these 2 main objectives, we want to go deeper and we want to have a higher increased productivity. We definitely want to have stable production results. Of course, reduction of production costs is an objective. And as much as possible, we want to have a simplified production process. It's also very clear that whatever we do in the start will definitely have an impact in the following steps of the production process. So this best start in life is really critical and fundamental for the success of the production in aquaculture. And if quality is decided very early, and when we look at the hatchery, the marine hatcheries are a very complex environment, and there's a lot of parameters that play into this quality. We definitely need to start well in terms of egg quality, and there's a lot of impact from broodstock conditioning and broodstock nutrition. It's the quality of the eggs will greatly vary and would greatly be impacted on how we have our broodstock and how we feed them. There's always, of course, a big impact in genetics, and there's a lot of parameters to do with environment. Here I'm including abiotic and biotic parameters, so from temperature, salinity to pH, light, to also how you stock the fish, what are the densities, if you put algae on the tanks, how you manage your water flow. So there's a lot of environmental parameters that will be critical and will play an important role on this quality. And then there's feed and feeding, of course. And focusing a bit on feed and feeding, when we think about hatcheries and we think even on starting new species, we look at nature and we think and we we look on how do they feed in nature. And there was a big step forward, of course, already many years ago, that when nature became industrialized production. So when we could start to produce artemia, rotifers, algae, and even more recently, we have copepods available. But the next step forward is definitely with feeds, with inert feeds. And talking about inert feeds, there have been important developments. So the larval feeds have evolved dramatically. They have done so nutritionally because mainly there's a much better knowledge in terms of larval requirements. Things are evolving. There's still a lot of things that we don't know, but there's a lot of development within the last year, especially in the balance of the micronutrients. But there's also a lot that has been developed on the physical parts. We went from feeds that the larvae would not even look at them because of the way that they would behave in the water, the way that they would move, the size and the shape, into where we are now, that we have developed methods to control all these parameters, and we have made the feeds evolve, and also evolve in a way that we can standardize them and have them stable with the same type of physical characteristics. And this has actually allowed to get to the stage where we are today, that the larvae do eat from mouth opening. So all of this has been overcome, and the larvae can actually eat. And when we're doing so, and we're checking if the larvae do eat, we've done quite a lot of different trials through different species. And what we have seen, and what we want to share with you today, is that not only it's possible to feed from mouth opening, but it actually produces better results. So we want to share with you early co-feeding strategies with Larviva ProStart. And what do we think that it does promote? We think it promotes a better biological performance. So here we're talking about higher survival, growth, and deformities, as we mentioned before as one of the objectives. But we're also talking about a lower CV. We're talking about promoting that it will be easier and faster to wean the larvae, and thus reduce the dependency on live feeds. And all of this goes hand in hand with what we said it was the objectives of the marine hatcheries, and also the main one, which is actually produce higher quality juveniles that will make a bigger impact on the grow out stages and will perform better. So let me show you a few trials that we have done across species. Let me share with you the results and why we think this is so interesting. Starting with bream trial, this was done in commercial conditions, so in a commercial hatchery in Greece, and we've done it. This work and the next trials are also are published. You have it, the link on the bottom. Karakatsuli Iol. This was done in collaboration with the Agriculture University of Athens, and we've done it in hatchery conditions in big scale. These were 18 cubic meter tanks. And we have compared starting feeding with ProStart on day 4 compared to day 15. And what we have seen in terms of survival and growth, they were equally good, but we have seen a big impact in terms of total deformities. In all the graphs that I will show you, you will see the light blue, it's the early co-feeding, so from mouth opening. And you can see here the impacts that the total deformities have reduced a lot. And you can see also the size, the uniformity in size. So the CV is much lower when you start feeding earlier. When we did these trials, the other thing that we wanted to understand was if there was an impact on the development of the digestive system. So we have analyzed quite a few digestive enzymes at the university. And what we have seen is that the development was normal, but there was a response to the early introduction of a higher protein level with the dry feeds in the beginning. So you would see that in this case, I'm showing the trypsin, but the chymotrypsin is showing the same trends. it responds. So it means that the larvae, not only you can see on the gut content that they are eating, but they are responding also in terms of digestive system. Going into red bream, different species, again in commercial conditions, in another hatchery in Greece, we have tested starting to feeding at day 3 versus day 22. I have to say that these are the normal usual protocols for the hatcheries. What we have seen was the survival was equally good in the two codes, but we have seen a difference in growth. And here, starting from very early on, we could see a trend that the fish that were co-feeding earlier from mouth opening were actually growing faster. And this growth was getting more visible towards even the end of the larval rearing. We could see exactly the same trend as deformities is concerned. So lower deformity rates when we start co-feeding from day 3. And again, the same trends in terms of the digestive enzymes and how the digestive system is responding to this early co-feeding with ProStart. Moving on to a different species, to sea bass, this time also in Greece, also in commercial conditions, so in big scale trials. We have done comparing starting feeding at day 7 versus day 25. And again, we have seen survival and growth were equally good. One of the concerns that we had and we monitored closely was if there would be an impact on the swim bladder inflation, since we were going to put the feed earlier on the water, and there wasn't. We still don't have the results of the digestive enzymes on this one, but we have seen again and consistently in another species the reduction of the commercial deformities. And we have actually, this was not planned from the start of the trial. When we moved the fish out of the larval tanks, the hatchery was pretty full and the tank availability and space availability was reduced, and we had to move them into very high density that needed to have a lot of water flow, so with a high current. And we know that when we put the fish in these conditions, there is a higher well, you will get them with hemolordosis basically. And what we've seen is that even if these fish were put in the same conditions, we've seen that the fish that were co-fed earlier from mouth opening actually had a lower rate of hemolordosis development. So it takes later on, and the quality of the fish actually goes on apart from larval rearing. Then we wanted to do also this in more controlled conditions. We wanted to follow the level of artemia that we were actually putting on the tanks. And we did another 2 trials with bream in semi-industrial conditions. So this is in smaller tanks, in this case in 400-liter tanks. And we compared feeding from start on day 3 to day 15. But with a very low level of artemia. We have used 12 kilos of artemia per million. And we have seen a higher survival. We have seen that even with this higher survival, meaning a higher density on the larval tanks, we still had an increase in growth. So we had a higher average weight, and we had a reduction also on commercial deformities. We retested this concept, but this time at the higher inclusion level of artemia, which is more realistic towards what is used commercially. So we went to 25 kilos of artemia per million, again comparing introduction of the larvae ProStart at day 3 versus day 15. And the same trends happen. We see lower commercial deformities when we start at open mouth. We see higher survival as well. And of course, when you look at all this increase and you think of all the trials, it really means that the impact is going to be on production costs. It's obvious that if you increase the biological performance, you will have an impact and the reduction of the production costs in million fry produced. So this is really important. So going a bit back, what did we say? We were talking about higher survival, higher growth, and especially what we've seen consistently was lower deformity rates in all these trials. We had seen lower CV, and we have seen on the trials really how easier and faster we can wean this fish. In most of the trials, this weaning was really anticipating because the fish were not even taking, they were not even keen to eat any more Artemia. So it was a much faster thing to do. And in some of this, what we've done as well is taking this larvae and carrying it on and following in terms of growth, the groups isolated. And we have seen that the growth were on the trials that there was already a difference in growth. This difference was maintained. And even on the trials that there was no difference in growth during the larval stages, we can see that further on at the nursery stages, then there was an impact, and the larvae that were co-feeding earlier had a higher growth. So why do we think that this is actually happening? Well, first of all, we have worked a lot on the nutritional and the balancing, especially of the micronutrients on the ProStart formula, and this means that this is actually it's formulated to fulfill the early requirements of the marine larvae. And it means that when you give them earlier, actually these larvae have their nutritional requirements met earlier. And as you know, when we use live feeds, even in the most stable productions, in the most stable hatcheries, even with the higher end enrichments, there's always a fluctuation in the nutritional profile of the live feeds. So what happens when you actually give this co-feeding earlier is that you buffer these fluctuations in live feed, and you are able to give a much more stable nutrition to the fish larvae. There's a big factor in the inclusion of the bactocell. We have in the larvae of ProStart the inclusion of a probiotic, and this probiotic, if you actually feed at the initial stages when they open their mouths, it's like having an open door, because the larvae are not colonized yet in terms of the gut. So giving the bactocell immediately from the start, you're promoting an earlier colonization of the gut, and you are promoting as well a better assimilation, a better digestibility of the micronutrients, especially on the minerals. And this has proven to have a big impact on the reduction of deformities. Of course, that with the feed, you can have a much more simple and standardized feeding, but you also are taking out some of the pressure on the live feed production. So you don't have, when you're in peak production, there's a lot of pressure to produce a lot of quantities of Artemia and rotifers, and the more pressure that you put on the systems, the more likely it is that you will have problems. If you have this buffer and if you can reduce it, you can much better also have this standardized and have better results. Of course, as well, that we are changing also the way that we are managing the microbiology and the microbiome of the tanks by putting less live feed, which is one of the big carriers of Vibrio, for example, that we put on the tanks. And it is a lot more buffered in terms of microbiology management. And this easier and faster weaning is not only what is promoted, but it's one of the reasons why, because it no longer you have this problems of some of the larvae are feeding, some are not, these mortalities on weaning, because the earlier you introduce, actually the easier it is to go through this phase. So in a nutshell, and as a take-home message, what would we like to say? First of all, it is possible to feed from mouth opening together with live feeds using live approach diets. These early co-feeding strategies promote a better biological and economical performance. We can get a much simpler and standardized off processing, off production and on results. And it is important that whatever we do in the beginning during the early development will have a significant impact on the next stages. So we really need to give our larvae the best starts in life. And that's it for today. Thank you. I'll stop sharing.
Thank you, Joana. Great presentations and great results. Our next speaker is Steven Debono, Product Manager of Fish Hatchery Product for INVE Aquaculture, part of Benchmark Holdings. He has been working within Marine Fish Hatcheries for more than 20 years. During these years, he held various positions from technical support, sales, and hatchery management. For the past 8 years, his activities are focused on contributing to developing products and protocols for marine fish hatchery production. The title of his presentation is Nutritional Solutions, Simplifying the Production of Quality Fish. Steven, hand it over to you.
Thank you. Thank you, Lucia. So, good morning, good afternoon, and good evening to all of you around the world. Today, as Lucia said, I would like to present you our nutritional solutions that are simplifying the production of quality fish in marine hatcheries. I am the product manager for INVE Aquaculture, which is part of Benchmark group, which is leading biotechnology company with a mission of sustainability in aquaculture. Our focus is on 3 complementary divisions: genetics, health, and INVE, which is the advanced nutrition part. And it's critical for aquaculture in order to achieve a sustainability across the production cycle. We are bringing specialized solutions to our worldwide customers in over 70 countries. But today, I will focus more on the hatchery part, where it involves a lot of work, and it's like an orchestra, actually. You have many players that require synchrony, attention to detail, and practice to create a wonderful symphony. And this doesn't come that easy. And of course, how we can support you to achieve this symphony, which sometimes it's not so straightforward. Of course, from our side, we can provide you with efficient and quality nutritional solutions. That is, products that achieve improved results in a more predictable way, accompanied with protocols fine-tuned to local conditions in order to maximize the performance of our products. Since the last time we met in this forum here, we have presented our new rotifer substitution diets that we are targeting to reduce by at least 50% the rotifers. The results have confirmed a major milestones in marine larva rearing, as we have worked this last year with many marine fish hatcheries in working on rotifer substitution and simplifying the production of the quality fry, reducing the running costs in the rotifer units, and all in all, improving fry production and performance. So today, I'm going to give you just a summary of 3 commercial experiences we have done this year in 3 different hatcheries that are producing more than 25 million fry, representing different protocols and local conditions, of course. The species involved were Sparus aurata, which is one of the major consumer of rotifers in marine fish hatchery production. The larva rearing volumes were between 10 and 13 meter cube. And in each experience, we have done either 3 or 2 tanks per treatment. Every time, of course, these values were compared to the control, which is the standard operating procedures of the hatchery. If we go into the protocols, our main target was to reduce, by at least 50% the rotifer consumption in the hatcheries. We apply a start feeding from day 4 and 5 days, depending on the temperature and at mouth opening, and the larval density of at least 100 to 120 larvae per liter. On the other side, the products that we use were Natura Pro and Excel. The rotifer feeding has been finalized by 24, but by at least reducing on a daily basis more than 50% of the total rotifers that they normally give. And the Artemia quantities were kept the same in order not to have any effects that could come from the Artemia part. Now, if you will look at the results, we will focus on the morphological development, on the survival, and also the growth and the stress tolerance or robustness of the fry. All the results I'm going to show you are at post-weaning, and we can start with the first result, that is the evaluation of commercial viability of the quality of the fish that we have achieved. And if we see the 3 treatments, in Hatchery 1 with Natura Pro and Excel, we reached 83% of good quality fry, and is similar to the control. Whereas in the second hatchery, we reached 97% with an improvement of 3% on the control. And in Hatchery 3, we went up, improving the quality of the fry by 13%, reaching 86%. So all in all, we see a positive trend in quality fry compared to the controls. If we go on survival rates, in the first hatchery, we have achieved slightly less survival rate, but still very high, when we see that the control was 58% and the Natura Pro and Excel were 53%. The other 2 hatcheries had an improved survival rate. From 32%, we went up to 38%, and the last hatchery went up to 41% from 33%. So in terms of survival rate, we see also very nice improvements when we introduced the dry feed from mouth opening, and also reducing by at least 50% the rotifers. Also, one has to see, we achieved also a more homogeneous survival with Natura Pro and Excel. An example of this is Hatchery 2, where here we see the survival rates of the 3 tanks with Natura Pro compared to the control, which is much more variable in terms of survival rates and performance. Regarding growth and robustness, for the growth, we have seen similar growth rates compared to the control, which has 50%, at least more than 50% rotifers provided. But what is very nice here is that we see an improved robustness and tolerance to stress, given also to the ingredients that we apply in the formulation, that is achieving a better robustness in the fry. Now, if we go on the rotifer consumption per million, we have seen that in Hatchery 1, we have achieved around 5 billion rotifers per million fry, in Hatchery 2, around 7 billion, and in Hatchery 3, around 9 billion rotifers consumed to produce 1 million fry. This is an extremely nice result, as we see that Natura Pro and Excel, in combination with the fine-tuned protocol, has performed effectively in different rearing conditions. And this is what we wanted to confirm. Actually, last year we have showed very nice results, and these results that are coming out, let's say, from our commercial scale hatchery, needed to be, let's say, challenged in the real world, in the hatcheries, in big hatcheries, where time and pressure on production are much more important than in small realities. And what we see here is indeed that we manage to provide a new solution that is really working very well in reducing rotifer consumption. However, one might ask, what is the impact on the production costs by introducing such a solution? And first of all, I would like to give you an overview of the estimated rotifer production costs, which is on average, we need 24 billion rotifers to produce 1 million sea bream fry. And the production costs that we estimate in total to produce these 24 billion is around 9,500 euros per million sea bream. As you can see, the cost distribution is 40% of culture and enrichment products, 23% of manpower, 20% is energy, you have 13% oxygen and water, and miscellaneous, which are, let's say, chemicals and other things that are required to maintain a proper rotifer culture production. So with this in mind, that we need 24 billion rotifers to produce 1 million fry, and we estimate that in order, if we take this number and we reduce it by 50%, 60% or 75%, we can end up with rotifer consumption per billion of 9, 7 or 5 billion per million sea bream produced. If we take into consideration this reduction in rotifer use, we can achieve a cost saving of 15%, or from 15% to 29%. This cost saving is taking into consideration all the production costs that you have of the rotifers that you have reduced, and you include also the feed up to weaning, that is a different feed, of course. And by this analysis, which is a great, let's say, result, it gives that partial rotifer substitution by Natura Pro and Excel is having a significant impact on the production costs. And also, never forget that we are improving the quality and the performance of the hatchery. And this we are contributing to a more sustainable hatchery production in various species that consume rotifers. So in terms of conclusions, the results show that we are improving in fry quality and hatchery profitability. So it's a solution to simplify and achieve a more predictable production. And then it's an important solution to further industrialize the process. And when I say industrialize the process, I think we have achieved really a lot these past 30 years. And by slowly industrializing the process, we don't want to lose on all the good effects that we have achieved so far. So with rotifer substitution diets, what you can achieve is that you are gaining a better performance, a better profitability. So from our side, from all the people that are involved in these projects, I would like to thank you all from us at INVE Aquaculture. And if you have further questions, you can always contact us. You can also scan this small QR code here, and you can get in contact directly with us. Thank you very much, and hope to see you soon. Bye-bye.
Thank you, Stephen. Great presentations and great results again. And for our audience, please don't forget to type your questions in the Q&A box. Our last speaker is Adriana Artiles, Business Development Manager for the Center for Aquaculture Technologies. The center is a leader in the field of genetic improvement for aquatic species. As part of the team, she brings her expertise and enthusiasm for both field and laboratory work to help clients worldwide find comprehensive solutions for their needs. The title of her presentation is "The Genetic Overview Reveals How the Captive Population is, and should be managed." Adriana, hand it over to you.
Thank you, Lucia, for the presentation. Thank you, everybody, for the patience to be listening. And the last one, I will try to use my time wisely. I hope you can see my screen. Can you confirm, please?
Yes, it's okay.
Okay. The title of the talk is the genetic overview to reveal how the captive population was, is, and should be managed. I want to thank at the beginning, sorry, we usually do this at the end, to my amazing co-workers that put together and shaped this service that we offer as part of the Center of Aquaculture Technologies. So the Center of Aquaculture Technologies has two headquarters. One part of the center is in San Diego, where we have the genotyping team and the gene editing team. And then the other headquarters are in the PEI, it's at Prince Edward Island, when we have the in vivo labs, biosecurity level 2 and 3, with all of the certification of the Canadian authorities. And the CIT, as Lucia mentioned, is focused on the application of technologies to improve productivity, efficiency, and sustainability in the aquaculture industry. And I'm very lucky to have world-class research co-workers. And as I previously told you, state-of-the-art facilities that are equipped to support diverse range of projects from discovery through final product development and testing to support the aquaculture industry. So we have, in the Canadian side, the health and nutrition team. We have even a new extruder where we can try new diets and formulations, and then we can try in vivo studies with the animals there, all type of aquatic animals, from oyster shrimp and fin fish. And then we have in the San Diego genotyping and genome editing, and the breeding team that I was belonging until recently, it's worldwide, helping clients to manage and go step by step for breeding plans. So we have everything under one roof, and that's what makes me so proud of belonging to this amazing team and contribute to the development of the aquaculture and the application of technologies. I will talk today about the genetic overview, and we use the raw data that is generated by the genotyping laboratory to create this service. So this service is provided by the breeding and genomic group. And we also carry out some other more complex services like the genomic selection, parentage analysis, genome-wide associated studies. And all this is done using this genotyping data that I will be talking to today. And in addition to that, of course, we have excellent teams to help with health diagnostic and challenge tests for animals in culture. And also we can carry out nutrition studies in vivo. And as I told you as well, we have the gene editing team that deploys innovations in all of the fields to improve breeding programs. And I think that my supervisor and colleague, Alan Tinch, gave a talk yesterday about this topic. Well, that's what we will exactly talking. I'm sorry, because my the previous speakers were talking about nutrition. This is kind of a different thing. We will be talking about genetics. And we will be discussing what is the genetic overview, why we need to use genetic overview, some of the applications, what type of arrays or species we can work with, what do we need from producers to carry out the genetic overview, and some examples and biological meaning of some of the parameters we use in the genetic overview, and of course, at the end, the conclusions. Well, what is the genetic overview? It's a compelling service to characterize populations in captivity that are usually managed by for commercial purposes. We use for genetic overview a set of softwares and tools, and we calculate and provide you graphs and tables to show the levels of diversity, the consanguinity, and the genetic structure in the population that we sample. At the very end, the genetic overview is a snapshot. It's like a picture of the current state of the population genetics characteristics. And then I will show you in details later, but in summary, we receive the samples, we do the inventory, we do the DNA extraction, and we do the genotyping and the analysis. And again, this is kind of something that sometimes we do not pay sufficient attention, because the genetic characteristics of the species, of an animal, of a plant, of anything that is in culture and we use for consumption, is not seen with naked eye. So you can control the quality, measuring the quality of the water, or you can manage the feed, as the previous speaker were talking about. But the DNA we cannot see naked eye, and everything that changed from the genetics point of view, and it can go through the next generation. So if we have a problem, we can be having the problem in the next generation. If we are doing great, our next generation will be excellent, and we will get the results from this next generation the best possible to the potential of the animal. So why then we need to use the genetic overview? Well, because when we do a breeding management, we do selection, and we do selecting breeding. If we don't track this, if we don't track this change in genetics, if we don't track in breeding, if we don't stand for a good diverse population, this accumulation of the inbreeding or relatedness, or this loss of diversity can eventually neglect the gains that we can have using a breeding program generation after generation. So the first things, as I told you, when we have a problem, the first step is to know what the problem is. Maybe we don't have a problem, maybe we are doing great, but that's part of the diagnosis we have to do as well. And that's exactly what the genetic overview does. The genetic overview will help you to know what is the problem, if there is a problem, and then will offer clues about how to solve it. Or by the contrary, if you're doing great and you are starting from a diverse population, it's kind of what the previous speaker said. So you have a buffer if you start from a diverse population, and you can lose a little bit if you have a big intensity of selection or something like that, but you're starting for a good foundation of genetics. And then you have to track how much are you losing in terms of diversity, or how much are you inbreeding your population, or by the contrary, if you're maintaining in good quality these characteristics or these traits in your genetic characteristics in your population. So that's why we have to do genetic overview, in addition to other good practices in the farm, of course. So some of the application of this service is, as I told you, we can monitor the inbreeding and relatedness and genetic diversity in a mass selection program. We can use it at the beginning of any new program of any type to assess the initial structure and previous management of the lines. So many times when we take a new breeding program, we don't know what happens before. What is the structure? How many lines do we have? Sometimes when somebody starts managing a breeding program, thinks, okay, I have 10 lines. And in reality, they have 3 or 2 or 1, and they think they have 10. So genetic overview can reveal that, or can reveal the origin, the ancestral background of the population's genetic ancestral background. Sometimes we take animals from a research facility, and then others from the wild, and we can know using the genetic overview what we brought, what is the quality of those animals from the genetic point of view. And of course, again, when another application is when we introduce a new line, maybe we have a breeding plan already, a breeding program in place, and we introduce a new line, and we want to evaluate how much different, how much better, or how much worse is this line compared to the ones that I had in my breeding program. And then we decide, okay, I will buy or I will not buy this new line. This will improve, or it gets worse my current genetics in the farm. And of course, in any ongoing management for breeding programs, different time points, when I want to see, well, I'm keeping my diversity, I'm getting, my population is in good quality from the genetic point of view, or is being inbreeding. So those are some of the applications for this type of analysis. Now, in CAT, we use arrays that use single nucleotide polymorphisms or SNPs. And we can have arrays from low density. We are talking here in the range of 100 markers, molecular markers or regions in the genome of the species, up to high density. So, and of course we have medium density. So more we screen in the genome, more information we have. But we can do genetic overview with any of these three types of arrays. So we are talking here again hundreds, here thousands markers, and here more than 10,000 markers, up to 50,000, 100,000, whatever we can get, depending on the size of the genome. And with all of them, we can carry out genetic overview. And every time we increase the density, we increase the applications. And of course, with the high density arrays, we can do more sophisticated applications like genome-wide associated study or GWAS, genomic selection, that we cannot do with lower density. But with lower density, with 192 SNP array, for instance, we can do parentage assignment, we can do genetic assessment or genetic overview that include the genetic diversity assessment, the inbreeding monitoring, the structure of the population, right? And that's what we will be talking about today. But we can do genetic overview as well with high density, and we will have by far more information. And we can also do right now, at this point, we have arrays for more than 25 species at CAT currently. And okay, this session is about finfish. We have for plenty of finfish. And if you don't find this picture, it doesn't mean that we don't have it. We have for many of them, not only the ones that are here, the snapper or the salmon or the yellowtail, sablefish, halibut, or cobia. We also have for sea bream, sea bass, and many other finfish species, tilapia, but also for oysters and shrimp. Panama is the most wide cultivated species, but we also have an awesome genomics team that can develop any new tool for species that are not in these that I'm telling you. And even we generate a tool for black-soldier fly, that is, this insect that we can use for formulation of feed. And we can use this tool to track inbreeding and diversity in population of this black-soldier fly. So if you don't see your species, we can still customize and create a new panel for you. Some of the co-authors of this work belongs to this genomic team. They are excellent co-workers and they are very smart and can generate this tool for you. So what do we need from the producers to carry out genetic overview? Well, we have to have the custody, the chain of custody record fulfilled. So we need to know what are you sending to us, what line the animal belongs to. With that, if we can associate to the physical tag, if we are doing something, we want something more complex, and we can provide you like Villeneuve matrices or something like that. And then you will collect the sample. So here you can see shrimp or fish, but also again from oysters, from black-soldier flies, wherever the tissue could be. These samples will be fixed in ethanol. Then you will pack the sample and ship to CAT, and we can send, of course, instructions for instructions to send the sample to us and the physical address and everything permits that you might need if you are sending samples from other place that is not the USA, the United States of America. I will give you a little visual aid to understand what are the single nucleotide polymorphisms and how, if we increase the density of the arrays, we can have more information. So here we have few SNPs, right? So let's figure out that we have a low density array, 192 SNPs, right? And then we have 2 individuals, so we can see that these 2 individuals are different, correct? So now when we bring this to a population and we have several individuals, we see that these individuals are very similar. So we have in this population a low diversity and high inbreeding levels, right? Everybody's similar. This population is not good from the genetic point of view. By the contrary, this one, right, we have here good diversity and low inbreeding. That's a simple visualization of what are we looking at the level of the DNA. And when we say SNPs, it's a base, nitrogenate base pair, can be adenine, thymine, guanine, cytosine, right? It's biallelic system, so you can have 2 of the variants on each individual. For parentage is useful as well, so I will just briefly mention. So we can have, let's say, this marker that we put red here as an example. In reality, those are not colors. Again, those are nitrogenate base pairs. And then we can exclude that these animals cannot be from those broodstock. The ones that could be from this broodstock should have this allele or this allele. So it could be this one, or this one. And we have a very comprehensive methodology as well to do parentage analysis, can do for clients, and we can do with any of the tools that I mentioned of this type of arrays for several type of species, more than 25 at this moment. So we are correct. Those are the offsprings related to these parents, and that's how we can also do molecular pedigree without the need of separating animals in tanks. We can mix all of them and we can track back to what parents this progeny belongs to. Now, we will see the example when we have a high density tool compared to low density tool, right? So here we have more SNPs, we screen better the genome, we have better visualization of and better coverage of the genome. And that's how we can do more sophisticated applications like the GWAS or genomic selection that can be used in breeding programs. So I would just choose, I have chosen 3 main figures or tables we use to describe the diversity, the structure, and the relatedness in the population that I will briefly share with you. So we have here a table that is the diversity parameters, and this has been done using synthetic data, so not real data from the clients, but simulation data. We have carried out this for several clients in different species, as I told you, but this is a hypothetic for populations and a hypothetic example that we did with simulation data using 192 SNPs in the array. And then we have 3 main parameters. One that is the best known are the heterozygosities that will tell you exactly this, if we have combinations of alleles in the population. And we also have the ratio of polymorphic loci. Higher this ratio, more polymorphic is the population and more diversity we have. So lower this ratio, we can have a non-diverse population, a population that will not have a lot of variation and not a lot of buffer. All the individuals, as I show you in the visual, will be very similar. And then we will not have a lot of room to choose and select from there. And the same, the mean number of alleles, it's 2 is the maximum per loci, as you know. So nearer to 2, it's better. Far away from 2 is worse. And the other one is the NAE estimator. It's a NAE diversity index. That is the probability that if I get 2 alleles from the population, they are different. So the highest population, in this case with the biallelic system, is 0.5. It's I take different alleles or I take the same alleles. Of course, if the population is less diverse, like population C and D, CC and DD, right? This probability will be lower. More we have this probability nearby 0.5, better. In this case, populations AA and BB are more diverse than population CC and DD. But this is not the, so this is like a general overview. This is the genetic diversity parameters. Again, when we start from the genetic diversity, from a diverse population, we have more buffer to choose. We have more buffer to lose on the path, adjusting the intensity of selection or something like that. And it is much better if we have a foundation of a good and diverse population. But this is not the only parameters we check. We also check an important one, that is relatedness. Relatedness will tell us when we compare pair to pair, when we do pairwise comparison of all of the individuals we have, we can do by populations or we can do overall. We will have like range that will tell us, okay, this pair of animals seems to be cousins or half sibs or full sibs. And then when we have normal distribution of these pairwise comparisons, like in AA and BB, and we have a mean nearby non-related values. So below these thresholds that I have here, around 0 point something, those are healthy and not inbred and not related populations. And then when we have the contrary, when we have like this type of tails in the distribution of relatedness, so we see there is something that is not good with this population as the previous one that was, as is in the previous table. So it's not diverse, but it's also related. So this is like a light, red lines. Is take care with this. These two are good. So it's green line. These two, we have a problem, a potential problem here. And also we have the structure that will give us with different calculations how many ancestral populations are contributing, how the populations or the lines are clustering. So sometimes this can give you clues. Okay, this line is the same, is diverse. You can combine them. You don't need to keep these as two separate lines because you're not managing these. You can adjust the labor, just merging the populations because they are bringing you the same or the contrary, or you have to bring a new one because you only have one line. So plenty of clues that can give you the structure of the population and in some of the history of how many lines are, from how many lines these current populations are coming. In general terms, the genetic overview is a powerful service that allows you to understand the current genetic characteristics of a population, describing the levels of diversity, consanguinity, and the structure of the population. It can be used, as I show you, for different applications and at different time points. And carrying out a genetic overview will help you to guide decisions in any kind of breeding program and for a great variety of species. And we can also offer customization of tools for non-traditional species that you want to try to culture, and then you can have good spawning or will have a low conversion rate, and you say, okay, this species is good for commercial applications. I want to see the genetics of my species from the scratch, and we can do it for you, and we can customize a tool for these non-traditional species. And I would say that it is very useful at the very beginning of any program or when you're introducing a new line to understand the story that the DNA has to tell you about how this population was in the past, how it's managed, and how we recommend you to manage. And thank you very much. I will be more than happy to answer your questions on the use of genetics in aquaculture. Thank you very much.
Thank you, Adriana. Thank you for the great presentation. And thank you all for the great presentations. I would like to ask all our panelists to turn on their videos and to begin with the Q&A session. So Joana, here's a question for you. how can the early feeding affect physicochemical quality of water, such as ammonia and nitrates?
Let me unmute first. The early feeding with inert diets, we're talking about very, very small quantities of feed that we're introducing in the early stages. Of course, depending on the feeds, it will have an impact on the system, and depending on how this feeding is managed, of course. Feeds with a lot more leaching, or with that the water stability is not very high, they will have a bigger impact. Also, depending how much do you feed per tanks and what is your water renovation, there will be an impact as well. I think for each different species and each different hatchery, there will need to be personalized protocols. And this is one of the important criteria that we need to take into consideration, is how we will impact the ecosystem and the water quality of the tanks.
Okay, thank you. Stephen, apart from these results, do you see other advantages in the application of rotifer substitution diets?
Thanks, Lucia. Of course, there are other things which are, let's say, on a more practical, but also from a managerial point of view, is that by liberating so much rotifers by at least 50%, you can increase your larval production by at least 50%, or else you can work more relaxed in the rotifer room and you have a better quality, and there you can also achieve a better fry quality in terms of the deformities as well. So you have many aspects there that give you a lot of flexibility and a lot of, let's say, removing the stress factor on our production people, especially in the larval rearing and the live food, which, of course, many hatcheries nowadays, I mean, are extremely good in producing rotifers. But on the other side, you know, rotifers are very strange creatures. 1 week they are okay, and the week after they start to sneeze. So yeah, it's really flexibility, I would say.
Okay, thank you. Adriana, can you tell us about the cost of your services?
Yes, so the genotyping for low density panels, it's about $10 per sample. And the genetic overview depends. So it is dependable of the number of lines, can be around 2,500 for up to 4 lines, and more than that, 4,000 if we are talking about 8 lines or many more lines. And for the high density, it's more complex. And then the tools, it depends. So we have for shrimp around $20 per sample. For other species, it's customizable between 25 or more than that, 40, depends. Or development of the panel, it's also requires a bioinformatic issues and sequencing of the animals, so it will vary. But in general terms, for low density, it's around $10 per sample.
Okay,
Plus the
good.
analysis.
Okay, good. Joanna, does your results apply for other fish species? There's a specific question on yellowtail kingfish larvae, if you have tested co-feeding that species.
The trends that we have seen cover a big range of marine species. Of course, species are different and facilities are different as well. So we have to be careful when applying things directly. I would believe so. We have not done any early co-feeding trials with yellowtail. We would be happy to do it, but a big part of this is it's to do not only with the feed, but with the feeding. So we're talking about creating a protocol that would suit the facility and would suit the fish species as well. what densities are we talking about? Is there 24-hour light, not 24-hour lights? What are renovations? What size of the tank? Hydrodynamics? All of this is quite important, and that's why on these strategies we believe that we need to be partners in terms of feeding, and it's so important. But I don't see why it shouldn't be tried with yellowtail kingfish.
Okay, good. Stephen, similar question for you. Have you tested Natura Pro in yellowtail kingfish or other species? You're muted.
Actually, it's not a real test. While we were doing these trials, one of the hatcheries had some yellowtail larvae and were not so great quality, let's say, in terms of broodstock and larva quality. But to all our surprise and to the surprise of the customer as well, so a much better behavior, and the larvae were much more active and eating well the dry feed. So I would say from an initial point of view, it can work, of course, but fine-tuning protocols and also understanding well the differences in the species. I mean, yellowtail is a fast-growing species compared to sea bream, and also the dynamics that you have in the larva rearing are a bit different. But I think there wouldn't be a problem at all. It's just a question of fine-tuning the protocols.
Okay, good. Adriana, how does genetic overview connect with nutrition? You're muted.
Yes,
okay.
Sorry. It's a good question. Everything in genetics is connected with phenotypics because we are the expression of what we have in our DNA for all of the species. So it is connected in the same way that health is connected with genetics. You have to have a good host, and you need to have a good variation in your population in order that this population reacts or respond to any change in the environment the best possible. So you can manage in a breeding program. Somehow you can make a select breeding for best FCR animals that you can choose from early stages. I know this is not a clear answer because you always have to design a good study, but knowing about the genetics of the population, you can somehow select animals that can have better feed conversion rate, or that can adapt to specific diets or conditions in the farm.
Okay, thank you. Joana, what are the gaps in larval nutrition you think need to be fulfilled to produce better and more efficient start feed for marine fish production?
The easy answer to that is some of them, we don't even know them. We know that there is still, in terms of nutritional requirements of early marine larvae, there is still, in some cases, a black box, and some things that we don't know yet because the trials are very difficult. These animals are really, really small and difficult to play with. But I don't think that the gaps are only nutritional. I mean, there is something, definitely, if we talk about the total replacement of live feeds, there is still something that it's missing on the dry diets, and something nutritional or something that it's bring, or that the Artemia or the rotifers bring in with them that we haven't been able to identify yet. But there is another part of it, and I keep on coming back with the physical part, because it has to be nutrition hand in hand with the physical part and the characteristics of the particles and the impact that this will have on the systems. And I think also the third aspect that will play a role, apart nutrition and physical, is also in terms of management and protocols, but it will be a change of paradigm if we actually go to total replacement and work with feeds. Then we need to start working on automatization in terms of feed deliveries. We need to start working about also automatic cleaning of the bottom of the tanks, a different management. So there's a few things that need to go into a different direction to get there.
Okay, good. Stephen, by reducing rotifor feeding with more microdiets, will this flow-on effect to using less Artemia? You're muted.
Well, what we have seen is that by having more survival rates, we have a better performance in terms of Artemia consumption. Now, having said that, when we see that we reduce Artemia in such a protocol, there are moments where the growth is not the same as in the control. So basically, we have customers who are using around 100 kilos in average per million fry produced of bream. And we have to be also very sensitive on this, that we want to achieve same growth, same quality as the controls. Now, having said that, of course, in the future, there will be room to be more efficient on the Artemia use and on the weaning strategies that you have. And this will be something that is being studied for many years now. But we need to know, we need to go slowly with this, because one thing is that we try low Artemia consumptions in, let's say, in semi-industrial conditions. And one thing is that we go with protocols in industrial conditions at real farm realities, which sometimes can give us a surprise. So yeah, slowly it can be done, fine-tuning protocols, fine-tuning conditions. It's not only Artemia, it's, let's say, the focus is more on improving on what we have right now and make it more simple.
Okay, good. Adriana, can you do a go even though you don't have a breeding program in place?
Yes, of course. And I would suggest at the beginning, if you don't have a breeding program in place and you want to start and you don't know where, the genetic overview is a good way to start. It will give you like this a snapshot that I was telling, and will tell you what are the genetic characteristics when you are starting from the scratch. So of course, I would likely recommend to do a genetic overview to start if you don't know where, for sure.
Okay, Joana, there was a question on the chemical composition of the feeds. Could you share which ingredients are you using or any other information you would like to share?
I'm not going to give the recipes and the formulation of the products. Sorry about that. But in general, what we're talking about is a high level of protein with inclusion of very highly digestible protein, and also of some of the small peptides, which at this level, at this larval stage, are very important. We also have on the lipid side, phospholipids are quite important, and some of the essential fatty acids, and especially the ratios between these fatty acids are important. Here we're talking about DHA, EPA, ARA. And on the micronutrients, if we're talking about vitamins and minerals, these are critical as well. They're very they're added in very small quantities, but they are extremely critical for the early development, and not only in terms of their levels, but also in the ratios. So the balancing of all these nutrients is quite important. Another thing, and I said on my presentation that for us is quite important, is also the inclusion the probiotic that we have on the feed that makes a big difference in terms of results. So not giving any formulas, this is the overview in terms of what we need to have inside.
Yeah, that's good enough, I think. Steven, Vibrio-related issues are a big headache for the industry. What is the impact of your new protocol in the Vibrio load in the larval tanks?
Yes. First of all, I would like to clarify something about Vibrio in general and the conceptions that we have with Vibrios. Vibrios are present in the hatchery atmosphere. It's not that a feed, any kind, live or inert, has Vibrio content inside. Then, having said that, it's very important that throughout the whole experiences that we have done, the fine-tuned protocol, the quantities were so little of feed that we have provided that were exactly comparable to the control tanks. So basically, in terms of Vibrio content, it was similar, and in some cases a bit lower. So there is no difference.
Okay, good. Adriana, what are the actions if the results are not good? Let's say if there is accumulation of inbreeding or low diversity.
It is a good question. So we have brain scientists that can help you with the decisions after we analyze the results. It will vary depending on the breeding program and the strategies you will give in place. So sometimes you need to introduce new flow of genes, buy new line. Sometimes you can do some breeding practice in the farm, merging lines, or reducing the intensity of selection, or recalculate breeding values. So there are plenty of options you can do after you analyze the results to improve the genetic quality of the lines and get the gains that you need for the production.
Okay, thank you. Joana, what's the FCR of Larviva in relation to the unit production cost of the fish? Hard question.
I'm not sure if I even understand the question. Can you repeat it, please?
What's the FCR of Larviva in relation to the unit production cost of the fish? I guess it's how the reduction in the FCR impacts the production cost.
When we talk about larval reading, I mean, FCR is not the first thing that comes to our minds. Of course, the feeds are expensive, and of course, the less you use will be the better. But it's not at this level really that there will be a big impact. You will always, when you do larval reading, you will always need to feed the fish and the tank. So there will be feed that is going to be lost, because as you do in rotifers and Artemia, for example, that you need to put certain amount of rotifers or Artemia per ml, because these larvae in the beginning are so undeveloped that they need almost to bump into the live feed. The same thing applies to feed. So you need to put a certain number of particles per ml. So there will be this tank feeding playing. So there will be waste, but really in these initial stages is not something that it is fundamentally important in terms of FCR. So I would probably leave it like this.
Okay, Steven, here's the question: is the complete replacement of rotifers and Artemia possible in sea bream and in other species with good growth and survival?
As we know now, today, no, it's not possible. You can bring some larvae alive for a certain amount of days. Yes, of course, but our primary objective is that we produce better quality fry than we have today. And until today, there is no feed that can replace completely live feeds, be it rotifers or Artemia. I don't know in the future. The trend is going to be there, of course, but I don't know if it's going to be in 5 years or in 10 years' time, because when you work with very limited amount of live feeds, you have other things that are influencing negatively our performance. And at the end of the day, I think nowadays there are standards, there are ways how to produce live food, which are quite simple. And I think the approach would be to make advantage of both solutions, dry diets and live feeds, to achieve a better performance than we have today.
Okay. Adriana, salmon farming is unthinkable without breeding programs, but sea bass and sea bream are low in adopting them. How can CAT help these producers? Can you make sex also determination for yellowtail kingfish?
I will answer the second one. That is easier. Yes, we can answer. We can know the sex using molecular marker for yellowtail. We have an universal marker that works for Seriola rivoliana, lalandi, and some other Seriolas, because it's for the genus. And the second one, for sea bream and sea bass, we have tools, and we would love to work with producers to establish this type of breeding program using molecular markers. And there are some other strategies. So we combine the strategies, measuring the phenotypes and using quantitative genetics, also with the knowledge of the genotypes. We can do more complex analysis, and we can do, as I told you, genomic selection that is like a higher level. But we can start simple, and we can do a conventional family-based breeding program with the help of molecular markers. And we would love to work with anyone with sea bream and sea bass in Europe, Asia, or any other place of the world. Happy to. Please mail me, and I will provide you more information on that.
Okay, good. Well, and that wraps up the Q&A. For those questions that were not answered live, they will be answered by email. I would like to thank our speakers today, Joana, Steven, and Adriana, for your time and the great presentations, and to our sponsors, BioMar and INVE Aquaculture. And thanks everyone for joining in. The recording will be available on YouTube soon. Our next webinar will take place tomorrow at the same time. Don't miss the 4th and last season on equipment and technology. And if you want to stay up to date on the latest news on aquaculture hatcheries, don't forget to subscribe for free to our publications on hatcheryfm.com. See you tomorrow. Goodbye.